This is lecture 34. It's our last one on anabolic signaling. And we'll pick up where we left off last time, talking about applications, but just the applications that we hadn't gotten to yet, which means nutrition, drugs, and supplements. First up, macronutrients, going to talk about nutrition, right? Carbs, fats, proteins, all three of these matter. We'll talk about carbs first, and we know how this one works. So for one, carbohydrate stimulates insulin secretion, and insulin is anabolic, signaling through PI3K. But glycogen also binds to the beta subunit on AMPK, and that inhibits it. So excesses of carbohydrate, that's going to be anabolic. It's going to help with anabolism. But depleting carbohydrate in the diet, this will associate with reduced insulin signaling and increased AMPK activity, which may be wise depending on your goal. If body composition and glucose regulation, if that's what you care about, then that may be a good idea. Although AMPK blocks mTOR and promotes protein degradation through both the lysosomes and the ubiquitin proteasome system. So, if protein anabolism is the goal, independent of health outcomes, then carbohydrate is an ally. All right, next up, protein. We know this one too. Particular amino acids are a trigger of mTOR. If your protein levels get low, if you have a protein deficient diet, then Reb and mTOR will dissociate. And no matter how much growth factor signaling you have, mTOR won't turn on. Protein matters. Deficiency inhibits signaling, and excesses promote excess signaling. And the major mechanism of amino acid sensing, this is the RAG proteins, which transport mTOR to the lysosome, the major work site of mTOR. And then also, some amino acids will trigger a modest release of insulin, leucine, glutamine, arginine, alanine. There are some amino acids that are going to get a modest insulin release. And then in some of these diagrams, you also see VPS34. It's a class of PI3K. So it makes phosphatidylinositol triphosphate, PI3P, as it's shown here, for phosphatidylinositol 3-phosphate. Leucine activates the kinase activity of HVPS34 uh, through increases in intracellular calcium, and through LRS. And then HVPS34, this produces PIP3. PIP3 recruits PLD1, PLD1 produces PA. So there are some other mechanisms through which particular amino acids can increase anabolism, but mainly this is RAGs. RAGs, that's the only thing you need to know. Um, and so if you're looking to eat the right amino acids, right, that are going to correspond to mTOR signaling, you have to look at the amino acid composition in the foods you're eating and in a variety of foods. And so methionine and lysine, these are both well sensed by the cell and can help promote mTOR. But in a lot of foods, you see they're really low, compared to meat and eggs and dairy products. So that's what these diagrams are showing. And the one on the right is showing essential amino acids. Potatoes have a good distribution of essential amino acids, but they just barely have any protein in them. So you'd have to eat a ton of potatoes to get a substantial dose of the essential amino acids. So choosing the right foods is important. One whole protein is not as good as the next. One source of protein is not as good as the next. Now, the timing of protein ingestion, that matters too, somewhat. So if you go exercise, you'll turn on muscle protein synthesis, but muscle protein breakdown is also happening. Remember this protein turnover concept? We have this balance between protein synthesis and degradation, and hypertrophy. That's just synthesis outpacing degradation. But until you eat protein, that's unlikely to happen. Hypertrophy is unlikely to happen. So in this study, they found that 20 grams of high quality protein, that's important, high quality protein, maximize the rate of protein synthesis for a 4-hour recovery period. And then eating more protein didn't increase protein synthesis beyond that. But this depends, that these numbers depend on a lot. The exercise performed, the body's doing the exercise, what was eaten before exercise. There's a lot of variables. So don't take those gram amounts as gospel, just the premise that if you don't eat protein around your workouts, you're not stimulating hypertrophy. You'll get muscle protein synthesis, but that's not the same thing as hypertrophy. You need to balance anabolism and catabolism, and you need an excess of anabolism to see the hypertrophy. So this is the same study talking about protein ingestion amounts and ingesting 20 grams every 3 hours resulted in more protein synthesis over a 12-hour period than eating 10 grams every hour and a half or 40 grams every 6 hours. But again, this is limited to the exercise performed, the body's performing that exercise, and they note their limitations in this study. And they also acknowledge, and this is really helpful that they do this, that other studies have found the ingestion of more protein resulted in more protein synthesis. In the study, they referenced 40 grams was better than 20 grams. So just use these values as a really loose guide, but acknowledge that hypertrophy won't happen unless you eat sufficient protein after you're done with whatever your workout is. And sufficient protein means high quality protein, and the amount varies based on the lot. But regardless of the amount of protein you eat, your sensitivity to ingested protein is going to linger for at least a day, probably longer. So if you eat high quality protein containing all the right amino acids, at any point in the next day or so, you're still effectively promoting anabolic signaling with it. All right, let's get into fats. Fats and cholesterol. Cholesterol is not fat, it's a waxy steroid alcohol. But in eating fat, if it comes from animal products, it's going to have a bunch of cholesterol. If it comes from, olives and avocados and other like vegetables, like none of which have livers, right? It's not going to have any cholesterol. But animal derived fats do. Anyway, with more cholesterol, you'll have more steroid hormone synthesis. And the primary effects of steroid hormones, we're thinking testosterone and estrogen, their primary effects are genomic, but they have secondary effects with transduction pathways, these signal transduction pathways. So what we're looking at here, this is a study we've seen before in which males went on a low-fat, high-fiber diet, and their androgens declined. And when they went on a high-fat, low-fiber diet, their androgens went up. Much more pronounced effects in the low-fat diets than the high-fat diets, though. But even if you didn't see, with nutritional modifications, even if you didn't see a difference in steroid hormone levels, the presence of lysosomal cholesterol helps signal mTOR localization through the arginine sensor. So there's lots of studies where like eating whole eggs has been shown to stimulate more protein synthesis than egg whites. With whole eggs, we see better mTOR localization, and that lysosomal arginine sensor, SLC38A9, it's not limited to arginine detection. It can sense cholesterol as well, and stimulate RAG, GTPase activation. All right, let's move on to some drugs and supplements. Starting with methylxanthines, this is the class of drug that includes caffeine alongside theophylline and theobromine. Theobromine, this is the one that's really abundant in chocolate. Not really in the fat of the chocolate, but if you can get cacao powder, it's pretty high in theobromine. It has caffeine too, but not much. It has more theobromine, and even less theophylline than caffeine. But theophylline, this is mostly taken as a drug. It does exist in some foods in really small amounts. There's some theophylline in tea, black more than green, yerba mate, and again, a small amount in chocolate. But all of these methylxanthines, these are non-selective PDE inhibitors. So non-selective means they'll have really diverse effects in the body. But what we've talked about, the version of PDE, phosphodiesterase, that we've talked about is 3B. That's the one in adipocytes in your fat cells. That PI3K and PKB activate as a part of insulin signaling. And if you block that PDE, you can facilitate more lipolysis, right? More fat catabolism, fat mobilization. And that's what non-selective PDE inhibitors will do, although the dose has to be high, and caffeine isn't as good of an inhibitor. Something else to keep in mind is that caffeine could activate AMPK, again, if the dose is like high enough, but there's no evidence that it's an mTOR inhibitor in the doses we're typically exposed to. But as an ergogenic aid, caffeine, but methylxanthines in general, right, as an ergogenic aid to strength performance, there's some utility. Your caffeine, your coffee, this has some utility. There is reason to include it in a pre-workout, concoction. The evidence is mixed, but it's compelling. Okay, amino acids, we've already talked about that, but I'll just touch on them again briefly. Some amino acids matter more than others in terms of mTOR activation. Uh, they increase the amount of GTP bound RAG-A that binds to Raptor, facilitates the interaction of mTOR with Reb. Now, protein translation can begin. You know the mechanism here and the mechanism of amino acid sensation, how we're sensing these particular amino acids. But there's something else that leucine can do. Is activate AMPK in the hypothalamus. Um, and so, or deactivate AMPK in the hypothalamus. So it can help promote satiety as well. Um, the leucine can. So leucine does have some diverse effects. And then in clinical populations that have poor mTOR signaling, leucine may have an even more pronounced effect. These amino acids that help signal mTOR can be even more robust in clinical population. So this study is looking at people with cirrhosis, which you commonly see elevated myostatin and corresponding sarcopenia, some muscle wasting. So how do we remedy that in these populations? And whenever you see elevated myostatin, you'll see impaired mTOR signaling and increased autophagy. So looking at phosphorylated levels of P70S6K, RPS6, and 4-EBP1, we see leucine working in the controls and really working in the cirrhosis patients. So it's effective for pretty much everyone, but it can be especially effective in clinical populations who are struggling with compromised mTOR signaling. Then one thing, though, that we should consider is that chronic changes um, in leucine ingestion is likely to elicit changes in cestrin expression. The body is constantly defending homeostasis and metabolic homeostasis, that balance between anabolism and catabolism. There's no exception here. So if you're always just gorging yourself on leucine, your cells will probably figure out a way to compensate, and expressing more cestrin is a reasonable solution. So even though leucine is just an amino acid, a component of protein, you're not going to OD on it. It might not be wise to try to OD like relentlessly, owing to probable regulatory changes. But now it's possible that there could be consequences of long-term excesses, um, other than just upregulating cestrin levels. There are some associations between blood levels of branched chain amino acids leucine, valine, and the likelihood of type 2 diabetes. I mean, branched chain amino acids are essential, right? You can't make them. So blood levels are either A, you ate them, or B, you broke down a protein that had them in it. But if you have a bunch of BCAAs, especially leucine, um, you don't degrade as much protein, and you're inhibiting, the catabolic signaling. So maybe you're eating these things. And the mechanisms for this effect have been proposed. Mostly it's a negative feedback loop. With a bunch of leucine on board, you're getting more mTOR activation. So obviously you're getting more phosphorylation of P70S6K, which is backwardly inhibiting insulin receptor substrate. So you're getting impaired insulin signaling, which is insulin resistance, which is type 2 diabetes. So maybe it's just a hypothesis. And some studies have found it to be more of a transient effect. So it's no reason to abstain from BCAAs. It's just something to keep in mind if you're constantly overindulging on leucine. And then one other a little point of commentary here. You'll see an occasional report about particular types of cancer, mostly pancreatic, and correlations with blood levels of branched chain amino acids. But this is probably explained by cancer-related tissue catabolism, as opposed to people just eating more branched chain amino acids and that causing the cancer. So this isn't something I would worry about, although robust mTOR signaling is not ideal in the presence of cancer. So if somebody already has cancer, inhibiting mTOR is a much better strategy for the management of already existing cancer. Now, as far as doses go, there is a dose-dependent effect. So this is a study that looked at muscle protein synthesis after cycling, where the exercisers either had 3.5 grams of leucine or about half that, and the higher dose corresponded to 33% more protein synthesis. Now, remember that it's helpful to signal mTOR through multiple amino acid pathways, arginine, lysine, methionine. So add in some of these other ones. And you can actually get other amino acids to bind to the leucine pocket on cestrin, isoleucine, and methionine, but you have to really go overboard with those doses. So getting in a normal dose of leucine, alongside a normal dose of arginine and a normal dose of maybe glutamine, methionine, and lysine, that would really help with mTOR activation, but there is a dose-dependent effect. Okay, HMB. This is hydroxymethylbutyrate. It's a derivative of leucine. Maybe 5% of leucine, give or take, gets converted to HMB endogenously, in your insides. But when taken exogenously as a supplement, it's been shown in the literature to have pretty pronounced anti-catabolic effects. Leucine has anti-catabolic effects too, but they work differently. There's some evidence of increased protein synthesis, but stronger evidence for its anti-catabolic effect. Since protein turnover involves both anabolism and catabolism, and hypertrophy can be achieved by manipulating either side of that equation, HMB is effective at promoting hypertrophy. Okay, so in this study, if you're an older adult being confined to bed rest for 10 days, that's a very catabolic environment. And supplementing with just 3 grams of HMB every day preserved lean body mass pretty effectively. So that's what this figure is showing. The HMB group didn't lose nearly as much muscle. And here's a look at the actual values they observed. And this is it's a good study. It's a randomized, double-blind, placebo-controlled trial. It's as good of a study design as you can get. And they were using DEXA to measure body composition. So it's not like they were using calipers or bioelectrical impedance or any of those huge margin of error assessments. As far as contexts in which HMB would be of value, obviously bed rest and bed rest-like circumstances, going into outer space or something, or you're an athlete and you're injured, so you can't train, and you're just trying to preserve the tissue you have until you can train again. And then also, if you're previously untrained and you're just starting a resistance training program, HMB may elicit greater improvements in strength and lean body mass for a time. Use it for that first month or so. Or if you change up your routine and what you're doing now, you're introducing novel stimuli that might correspond to more tissue breakdown, then you might find HMB working in that setting too. But if you're already well trained and well adapted to the program you're doing, and you're using HMB with the goal of promoting anabolism, the ergogenic effect doesn't seem to be quite as good there. As far as dosing goes, 3 to 6 grams per day, that's normal, divided into a few doses. All right, next on the list is trimethylglycine or betaine. This, what people would often pronounce betaine. So this assists with the methylation of stuff as opposed to phosphorylation. In this case, we're adding a methyl group, which is a CH3. So this is a methyl donating compound. And there's one really nice study that looked at PKB signaling in response to supplementation. I like this study in part because I know pretty much everyone on the author list. Jenna and I did our PhDs together, and everyone there does really careful work. And this is a placebo-controlled trial where the subjects, there's 12 trained males, spent 2 weeks on either betaine or a placebo. And then they did a washout period also for 2 weeks. And then they switched groups. Whoever was getting the placebo before, they're now getting the betaine and vice versa. And that lasted for 2 weeks also. So the 12 subjects served as their own comparison group. And then they measured growth hormone and IGF, both in circulation. And then in the vastus lateralis is a quadriceps muscle. In samples, they measured PKB and P70S6K. And here in these figures, you can see the post-betaine elevations in growth hormone. And as you would expect in the presence of elevated growth hormone, IGF, they did see elevated IGF also. Um, and if there's an increase in IGF, which they found, you'd expect to see increases in the phosphorylation of PKB and P70S6K. And they found both of those too. They also found a decrease in cortisol. Um, so this placebo-controlled study, it showed compelling evidence of betaine or trimethylglycine on anabolism through elevated growth hormone and anti-catabolism through reductions in cortisol. If you're curious to read about it, this is the abstract, which summarizes the methods and findings. I've highlighted the major findings here in the blue font. And here's the conclusion from that same paper. Again, compelling evidence of an anabolic effect and an anti-catabolic effect. It's modest in its magnitude, but significant in its probability. All right, moving on to phosphatidylserine. The diagram on the left is from the top article. The bottom article is just echoing that point, which is that resistance exercise causes the release of a bunch of stuff. And among that stuff is cortisol. And as we've already discussed, cortisol is super catabolic in a bunch of ways. And that's where phosphatidylserine comes in. It inhibits spikes in cortisol. So in the figures here, you can see plasma adrenocorticotropic hormone on the left. That comes from the anterior pituitary. It's the intermediate hormone in the HPA axis, the hypothalamic pituitary adrenal axis. The hypothalamus releases corticotropin releasing hormone, which causes the anterior pituitary to release adrenocorticotropic hormone. That's what they're looking at here. And then in response to that, the adrenal gland releases cortisol. And so on the left, that's adrenocorticotropic hormone, and on the right, that's cortisol. With the placebo, both of those hormones spike in response to physical stress. With phosphatidylserine, it doesn't eliminate either of them, but it prevents the spike. And even though these doses are, I mean, these are pretty small doses. It's hard to find a pill that's less than 100 milligrams. These findings hint at being a dose-dependent effect. All right, fast forward the world a few years, and the tested doses are considerably larger. A moderate dose here is 600 milligrams per day. So remember, the last study was 50 or 75 compared to a placebo. So this is getting into a more substantial dose. And you can see the methods here. They had 10 male subjects. It was double-blind, placebo-controlled crossover study. So just like the beta study, all subjects received both the placebo and the treatment and were compared to themselves. The exercise, it was 15 minutes of cycling, increasing intensity, and the researchers evaluated circulating lactate, growth hormone, cortisol, and testosterone. And you can see the cortisol levels were much lower in the phosphatidylserine group, even with the small sample, statistically significant. Nothing else reached significance, but testosterone was a bit higher in the phosphatidylserine group. Compelling magnitude, but the p-value is 0.13. It makes sense. This finding makes sense as cortisol blunts testosterone. And then on the right, we see a plasma lactate. No difference. So the implication here is that the exercise workload between the phosphatidylserine sessions and the placebo sessions were the same, but the growth hormone levels in the phosphatidylserine sessions were a little bit lower, not significant. P is 0.15, but there may be some effect there. But luckily, a supplement that you could take in addition to this one that also has an increase in growth hormone in addition to lowering cortisol. All right, moving on to arachidonic acid. You know what this is. It's 20 carbon fatty acid. It lives in your phospholipid bilayer, and it's a precursor to prostaglandins, which are the major cause of your delayed onset muscle soreness. But prostaglandins also promote anabolic signaling. So in this study, they found some improvement in muscle performance. The dark bars, that's subjects using arachidonic acid. The white bars are subjects receiving the placebo. Probably hard to hide a placebo effect, the soreness they feel. But they looked at the bench press, leg press, and Wingate and found better improvements in general in the arachidonic acid exercisers. Here are the methods. It was a pretty legitimate exercise program. I mean, it's just Monday, Wednesday, Friday, only 3 days, but real exercises. And they did it for 8 weeks, and they measured body composition using DEXA. So again, accurate assessment device there. And then looking at both muscle protein synthesis and lean mass changes, the arachidonic acid group did better. So there's evidence of effectiveness here. It's not a supplement that I would personally do. We talked about in the second block how inflammation needs to be well regulated. Too little or too much can get problematic in a hurry. For most athletes, this is probably fine. This supplement is probably fine. But for me and anyone else who would have reason to worry about inflammation, I'd skip over this supplement. Next up, phosphatidic acid. You know what this is. You know how it works with mechanical loading. Diacylglycerol kinase phosphorylates a diacylglycerol or a diglyceride. That gives us our phosphatidic acid, which helps activate mTOR. But if you just eat the phosphatidic acid, it doesn't do exactly the same thing. It doesn't work in exactly the same way. And I'll talk about that in just a minute. But this is the abstract of that study. They were looking at how much P70S6K was phosphorylated compared to how much was unphosphorylated in C2, C12 myoblast cells, an immortal line of mouse myoblasts. And then they also investigated humans exercising and assessed improvements in bench press and leg press. Same article here that we're looking at, passage from their introduction, just a simple narrative of PA, of phosphatidic acid, as a messenger molecule, which accounts for part of the mechanism through which mechanical loading turns on mTOR. And here they're talking about the difference between endogenous PA and exogenous PA, meaning you eat it. And if you eat it, you convert some of it to lysophosphatidic acid, LPA, and that signals the MAPK pathway. And they report that some of it can increase PLD activity. That's phospholipase D, which makes phosphatidic acid from phosphatidylcholine. You don't need to know that. Just know that the mechanism of action when you eat it is a little bit different from endogenous production through mechanical loading. And this is just showing the effect of phosphatidic acid supplementation on strength outcomes. Leg press on the left, bench press on the right, and total strength in the center. That's the summation of both bench press and leg press. I don't really like leg press as a assessment in studies like these because anyone can do 10 more pounds, upon their max. And then they could do another 10 pounds. And then if they wanted, they could add 10 more pounds to that max. So there just seems to be too much variance in capacity for my stamp of approval on leg press. But they did see changes in one rep max in strength, more in the PA group than the non-PA group. The asterisks, that's significant changes. Other studies have found positive effects of phosphatidic acid supplementation as well, but not every study does. This one we're looking at here didn't find any value to phosphatidic acid. It's on older adults, age 65 to 75, not very many subjects, and they were just doing unilateral knee extensions. It wasn't a good study design, from the perspective of exercise stress. It wasn't very good. But they did capture mTOR activity sufficiently. And so it was effective from that perspective. So here are their findings. Placebo, darker lines, rest and exercise condition. And phosphatidic acid, lighter lines, rest and exercise condition. 150 minutes post-exercise and 300 minutes post-exercise. So that's what we're looking at. And what they were evaluating was the phosphorylation of AMPK, P70S6K, PKB, MAPK, 4EBP1, and ribosomal protein S6. And phosphatidic acid doesn't fare well here. The results aren't good. They don't favor it. But that doesn't mean it doesn't work. This is just an uncharacteristic study. It could be sampling error, it could be the exercise population or the exercises performed. It's any number of variables to explain these findings. But phosphatidic acid tends to work pretty well in the literature. But this study had a much better resistance training design. It was a randomized, double-blind, placebo-controlled trial, 15 subjects with a history of resistance training, completed 8 weeks of that training. The average age was 23, give or take a few years, and they used ultrasound to measure the muscle thickness. And they didn't really find anything either. There's nothing compelling here with oral phosphatidic acid. So there are reasonable studies that fail to find an effect with phosphatidic acid, but the majority do see benefit, and there are known mechanisms for how it would exert its anabolic effect. Okay, let's talk briefly about a supplement that's really effective, that really works, and that's creatine. The creatine kinase reaction, creatine phosphate donates its phosphate to ADP, so you get your ATP back and you consume hydrogen ion in the process. Now, creatine works well through increasing the total content of phosphocreatine. Your performance in very anaerobic tasks will be enhanced accordingly, according to how much phosphocreatine you have on board. But creatine has other effects too, and this includes increasing the number of myonuclei. So remember that a skeletal muscle cell, can only grow to the extent of its governable domain. So if you want to grow more, if you want to grow beyond that, you need more governments, more nuclei, and creatine can assist with that part. All right, moving on to catabolic supplements. Anything that's going to promote AMPK activity is going to have a catabolic effect, in addition to aerobic enhancements. And so that's why people are doing these AMPK supplements in terms of sport, performance and human performance, whatever is the aerobic enhancement. Now, it's not really gene doping, as it's claimed to be. So look at item 3 in that list of prohibited gene doping agents. I quote, the use of agents that directly or indirectly affect functions known to influence performance by altering gene expression. And then they list ICAR as an example. We talked about that one. It's a AMP mimetic. Does it change gene expression? Yeah, right. But so does leucine, right? And so does a banana. If you eat a banana, you're manipulating gene expression through insulin. So I don't like statements like this. Yeah, they're just clearly not written by physiologists, but supplements that stimulate AMPK can be powerful ergogenic aids. It just depends on the sport. So here's a list of activators. I mean, I have 5 of these in my supplement cabinet now. You can get them from grocery stores, um, like quercetin and resveratrol. I mean, those are just polyphenols. You can get resveratrol in nuts and grapes and berries. You see curcumin on there too. That's a polyphenol and turmeric. But also on this list is drugs like metformin, which is way more powerful. Overall, though, this is a good list of compounds that can help activate AMPK indirectly. When you see the molecular target being complex 1 of the mitochondrial respiratory chain, that's what it says. That's a target. Potentially many other targets for some of these compounds, but that's a big one. So, for example, metformin also inhibits AMP deaminase. So, AMP clearance is compromised. So, AMP builds up. But complex 1 of the electron transport chain, that's a common target. And that's not catastrophic to inhibit. Um, like cyanide inhibiting cytochrome C oxidase, that's going to harm you. I mean, that's bad. Here, inhibiting complex 1, that's fine. A little complex 1 impairment just means the body has to sing a little harder for its supper. It's still fed, right? It still works. It's just a little harder to get that ATP regenerated. And then here's a list of direct AMPK activators. In the target subunit, right? You can see the AMPK subunit it binds to. So ICAR, that's binding to the gamma subunit. So that means it's binding where AMP binds. And so it permits activation by LKB1 without needing a bunch of AMP on board. You don't need a ton of energy stress if you just mimic it with ICAR. Mostly you see these compounds binding to the beta subunit, though. That's the one that glycogen would bind to. And then once AMPK is on, you know what it does. Okay, so let's look at some what I'm calling a maybe bother. Those previous ones, those previous supplements, if they are oriented around your goal, go for it. For this is a maybe bother taking it. And vitamin D, lots of conflicted study results here. Some find a little bit of benefit, some don't find any benefit at all. That said, like if you're a troglodyte who prefers the back half of the cave, to the open sunlight, vitamin D, that's going to help. As far as its functions, it's closer to a steroid hormone than a standard vitamin. It's cholesterol derived. Like we talked about androgens binding to androgen receptors inside of the cell, and then the hormone receptor complex binds to androgen response elements in the DNA. Vitamin D is similar. Um, Calcitriol, this is the active form of vitamin D, and that binds to the vitamin D receptor, which then binds to a vitamin D response elements in the DNA. And so again, it's closer to a steroid hormone, but its relationship with strength and hypertrophy isn't clear. It's man, vitamin D is helpful, and go ahead and take it. But for this particular reason, maybe. All right, getting into the probably don't bother supplements in terms of muscle mass and strength, right? Omega-3 fatty acids, you should probably be taking these unless you're getting a ton of omega-3s in your diet already, fish and like fatty fish. But the reason you take them isn't for skeletal muscle hypertrophy. The evidence there is really weak. CLA, this is another fatty acid. It's actually a trans fat, this one, which we think of as bad. Trans fats are bad, but this is a natural trans fat. I mean, just eat some grass-fed cows and you'll get some CLA in your diet. And it's taken for a lot of reasons. Mostly people are taking it for the claim that it reduces body fat. That claim usually holds up in the literature, although the effects are modest. They're really modest. But the strength and muscle mass benefits are pretty flimsy. If they're real, they're much more modest than the fat loss. And it's not really worth bothering with CLA for this particular goal. All right, another don't bother, citrulline. This one, it's occasionally recommended to help with muscle mass and strength, but the literature just doesn't support that recommendation. All right, another don't bother, antioxidants. We know that a small increase in reactive oxygen species has an anabolic effect, while a large increase will have a catabolic effect. There is uh, complicated interactions, a complicated relationship with mTOR signaling here. Um, and we do see oxidative stress with advancing age and with disuse atrophy. But if we're looking at a healthy person, healthy populations, antioxidants are not going to be helpful for strength and hypertrophy. Now, if there are pathologies at play, maybe. But even in sarcopenia cases, the evidence isn't really clear. For healthy folks, though, if your goal is hypertrophy or strength and power, don't bother with these. Another don't bother, Tribulus. This was a big one around, I don't know, 2000, the year 2000, so around there. Lots of people started taking this to boost testosterone and just championing it like it's a natural alternative to anabolic steroids. But the research hasn't validated those champions. Tribulus doesn't really seem to work, so don't bother with that one. Ornithine, I'm putting on the don't bother list too. There are a couple of studies that have shown some support, but they're weak studies and you won't find them on PubMed. So here, what you see is citations 317 and 318. Unless some compelling support begins to emerge, don't bother with this one. And that's the last supplement that we'll talk about and the last of our applications. And this concludes the last of our mTOR lectures, our anabolic cell signaling lecture. So here are the review questions for this one, for lecture 34, the topics to pay attention to, and I will see you in the future.